So as we go over the topic of cell signaling, what I want to focus on now is this idea of protein-protein interaction. Things have to touch each other. It's a very physical process. So again, when we look at these signal transduction maps, we can see that these proteins in the cytoplasm or signals at the exterior of the cell and the exterior of the cell have to interact. They have to touch each other. Right? They have to touch their receptor. The receptor changes shape. It touches something in the cytoplasm, which then now has been activated. And it goes off to activate something else. It has to bump into it. And again, all of these things are going to be concentration dependent. Right? So there's no intelligence in a protein that says, now I must seek out the next partner in this chain. No, it is random. And so the cell has to maintain all of these proteins to a certain extent to maintain the effectiveness of its signal transduction pathways, and ultimately this means the effectiveness of its ability to process information, okay, and respond to it. So the themes that I want to focus on today are the idea of protein-protein interaction. These things have to touch each other, right? And then form and function, right? It's the shape of a protein that's going to allow it to interact with something else in a specific way to cause a result. Phosphorylation is another theme, and we're going to look at that in detail. It's going to be a very, very common on-off switch that cells use to turn proteins on or off in these cascades. And then again, this idea of the protein changing shape to be either in an active shape or an inactive shape, and that we can control that. So there's almost two modes for every protein that's kind of involved in these pathways, one of which is going to move things forward, and the other is going to be inactive. Right? So let's start with protein kinases. Now, a kinase is a term that you just got to become familiar with. It may feel awkward to you now, but you just need to use it again and again. A kinase basically transfers a phosphate onto a protein. It phosphorylates proteins. And we're going to see kinases again and again in many different forms. Phosphatases are the opposite. So a protein phosphatase removes the phosphates from a protein. So now, all of a sudden, when you talk about this pair of proteins, you can see it working in tandem as a switch, right? Phosphorylation and dephosphorylation can act to turn on and off proteins. So we oftentimes will talk about what's called a phosphorylation cascade, a protein that is phosphorylated, it turns it on, and then it in turn phosphorylates other proteins, which in turn phosphorylate more proteins. And we'll take a look at that a little bit later. Now, again, this idea that a kinase will phosphorylate a protein, add that phosphate group on there, and it can be dephosphorylated. This isn't so much about energy, because we've talked about ATP and phosphorylation and dephosphorylation kind of as an energy mechanism. Here, we want to think of it much, much more as a switch. So that protein being phosphorylated now is in an active form. It changes its shape, having that phosphate group on there. Dephosphorylated changes its shape as well. Whether phosphorylation turns it on or off is dependent on the protein. So don't think that if it's phosphorylated, it's now active, though that is in many cases what happens. You can also dephosphorylate a protein to activate it, so keep that in mind. This is protein dependent. Now, again, coming back to the theme that form and function is so important in biology, what do we have here? We have molecules interacting. And so what we call the signal molecule or the small molecule interacting with a protein, we call it a ligand. So the interaction or the relationship between a ligand and its receptor, or the protein that's it's going to bind to as part of the cell, is very, very specific in many cases. Right? So you can think of an antibody that is highly specific to its antigen. This is a ligand-protein interaction, a ligand-receptor interaction. So when that ligand binds to its receptor, it can change the shape of the protein. And when it changes its shape, that can activate other proteins. So most receptors are found in the plasma membrane, though we'll talk about alternative receptors in just a few lectures. But when I interact with something on the outside of the membrane, it may change the shape of the protein on the inside of the membrane. And that has now conferred information from outside the cell to inside the cell. Now, when we start to look at proteins, what we're going to see is another level of structure that we haven't talked about yet. Go back and review protein structure. Primary structure, secondary structure, tertiary structure, quaternary structure. But when you look at enough proteins and characterize their structures, what you start to see is what is called supersecondary structure. These are motifs, such as you would talk about motifs in music, a recurring series of notes or tune. You'll see a recurring structure in proteins. And these can be great hints as to the function of the protein. So common shapes, combinations of loops, helices, or sheets, which we know are secondary structures, but we see them gathered together in a specific form. We call this a super secondary structure. They're often very much involved in binding ligands or cofactors, transporter proteins, or interacting with DNA. And the more you talk about this stuff, the more you see it, the more you'll recognize this. So I'm going to give you just a few examples so you can kind of see it in your head. But here's what's called the EFhand calcium binding motif. So again, this isn't the whole protein. This is just one little part of it. But there are many, many proteins that bind calcium that you'll see this very common motif. This helix, this alpha helix, a little bit of random coil beta sheet action, and another alpha helix kind of shooting off the bottom there. Now, I don't know who looked at this and said, well, that looks like a finger and a thumb and a hand. I wouldn't have gotten there myself, but that's what they call it. And so you can see now how they have drawn it now with the corresponding colors to show you how this kind of folds around a calcium ion to bind to it. Right? So we call this an EFhand. Beta barrel, another very common structure. We oftentimes see these in transporters or even in toxins. As toxins insert themselves into a membrane, they make a hole in the membrane, and that's how their mechanism works to be toxic, by dissipating the membrane potential. Zinc finger, very, very common motif. This one binds a zinc ion and is involved many, many times with interacting with DNA. So you have this part of the protein now that looks like a little finger, and it can fit right into the major or minor groove to read the DNA, again without separating the double helix. Go back and review that concept as well. But these super secondary structure motifs are good indicators of the function of a protein. But again, the big idea here when it comes to cell signaling is that when we bind to something, the shape of the protein can change, and often changes. And so for here, as an example, we have the insulin receptor, and it undergoes this conformational change when it binds insulin. And you can see on the far left here, unbound receptor, which then binds this blue insulin, and it changes its shape. It doesn't just change its shape on the outside to grasp the insulin, but more importantly, it changes its shape on the inside of the cell and activates the functionality of this receptor. This is going to lead to passing that information on through phosphorylation to subsequent proteins. All right? And just an idea, this is from a website on cell biology. So they talk about the signaling complex gives rise to a wide range of intracellular signals, a wide range of downstream effectors involved in a wide range of cellular responses. This is just one piece of informational input that the cell has to process. And how does it process it? It processes it with these large interacting protein networks. All right? So this is the level of kind of information processing, the computer level of the cell. So the more you study this stuff, the more I find it absolutely fascinating.